Hey everyone. So this is part 2 of the 3-part series on antibiotics for the biomedical science exam. For those of you who haven't seen part 1 yet, I suggest going back and seeing that before you see this video. It'll make a lot more sense. And for everyone else who has seen the first part of the video, I just want to quickly review the take-home messages from the first part of the video. So the first thing we established was a system for classifying bacteria. And if you recall me saying, don't get too worried about remembering where every bacteria every bacteria on this list fits at this stage. In the third part of the lecture series, I'll be talking about the main causative organisms of clinical infections that come up in clinical practice and in the biomedical science exam. So at that stage, it'll start to become very clear where these things fit and why use certain antibiotics. So I think that will be a penny dropping kind of lecture. I hope it will be anyway. But at this stage, all I want you to sort of remember is that the majority of the bacteria that we encounter in clinical practice are these ones, gram-positive cocci, or the rest of them are gram-negative rods. That's all for now. Remember as many as you can at this stage. But also remember that there are atypical bacteria that don't actually show up on the gram stain. And just remember, big ones that come up, particularly for the biomedical science exam, are mycoplasma in this list, right? And we said that there are 4 classes of antibiotics. And we went through the first class of them, the cell wall synthesis inhibitors. And today, in this lecture, we're going to go through the bacterial translation inhibitors, the nucleic acid synthesis inhibitors, and the unclassified bacteria, antibacterials. Just to specify which antibiotic, because I know there are a lot of antibiotics we went through in the first part of the lecture, I would say that you should know your penicillins very well. So know all the generations of the penicillins and know how they work in terms of their spectrum and their sensitivity to beta-lactamase. Know the generations of cephalosporins. Again, you use these all the time in clinical practice, and they love asking you what the difference between a first and a third generation cephalosporin can be. The imipenems, meropenems, although they are important drugs, and equally as trionam, although they are important, they don't really come up too much. So if you want to, at this stage, learn something really well, I would suggest it's these 2 classes. And vancomycin is very important to remember as well. Okay, so those are the main things to take over from the first part of the lecture series. Right, so today we're going to be talking about the drugs that inhibit bacterial translation. And this is quite a large group of drugs, actually. It includes, as you can see, the aminoglycosides, the tetracyclines, chloramphenicol, macrolides, clindamycin, and linezolid. Now, this is quite a large group of drugs. And so in terms of their mechanism of action, again, you can break them down into a common mechanism of action. And we're going to just talk about this definition down here in a second. But what I just want to say, just to sort of solve any confusion that you might have, a lot of the textbooks go into a lot of detail about the mechanism of action of these drugs in terms of what part of the ribosome they attach to, and what part of the protein synthesis process they actually inhibit. In my opinion, and in my experience of what's acceptable at the biomed exam and what's necessary and what the focus is, I don't think you need to go into that much detail. So that's why I would recommend talking about, when you ask about the mechanism of action of any of these drugs, to say that they either bind to the 30S or 50S ribosomal subunit and inhibit the synthesis of bacterial proteins. I think that's all you need to say. Now, which of these drugs bind to the 30S ribosomal subunit and which of them bind to the 50S subunit? There's this mnemonic, which is from a USMLE textbook, called buy at 30, sell at 50, right? And that helps you because the drugs that bind to the 30S subunit are at, so that's the aminoglycosides and the tetracyclines. And the ones that bind to the 50S subunit are the cell ones. So that's the Chlorampphenicols, erythromycin, and the rest of the macrolides. So that one doesn't quite work because erythromycin is one of the macrolides. But if you just remember erythromycin is a macrolide, it is erythromycin and all the macrolides bind at 50S, and the lincosamides such as clindamycin, as is mentioned above. Right? So that's an easy mnemonic you should remember to tell you which of these translation inhibitors binds to which ribosomal subunit. And in my opinion, again, I think the only definition for mechanism of action you need to remember is this one. This is going to be, as much as it is a biomedical science exam, it's a clinically focused exam, so they don't expect you to know the very intricate molecular biology of all these drugs. So let's start with the aminoglycosides. Now, the aminoglycosides is a special group of drugs, and what you should know is that the bacterial translation inhibitors are all bacteriostatic, in that they don't kill bacteria, but they simply stop their replication through inhibiting their synthesis of bacterial proteins. Aminoglycosides are special bacterial translational inhibitors because they're the only BTI, bacterial translational inhibitor, that's actually bactericidal. And the reason they're bactericidal is because they can actually cause a misreading of genetic code when constructing proteins, when the bacteria is constructing proteins, and this leads to the wrong amino acid being placed in a polypeptide protein, and that leads to a destruction of the bacteria. So aminoglycosides, just remember, it can come up that they're actually bactericidal, unlike the bacteriostatic bacterial translation inhibitors. Right, so that's written there. They're accumulated actually intracellularly via an O2 dependent uptake. So they require oxygen to be taken up by the bacteria. And for that reason, the anaerobic bacteria are actually resistant to the aminoglycosides. The main aminoglycosides that you would have come across are probably gentamicin, but you might have also heard of tobramycin and streptomycin. That's the one that we use clinically most of the times. Their spectrum is pretty good with the gram-negative rods. Yep. And pharmacokinetics-wise, you've got to be really careful with gentamicin because its renal elimination is proportional to the GFR, and major dose reductions are needed in renal dysfunction. And it's one of the main things we worry about with aminoglycosides' effect on the kidneys. So just as we were talking about, the main side effects of the aminoglycosides are that they are nephrotoxic and can lead to an acute tubular necrosis. They can be ototoxic, i.e. toxic to the ears. And then those are the two main ones. If you want to throw in another one, so they can actually cause a neuromuscular blockade. And the reason they can do that is because they can decrease the release of acetylcholine due to their initiation of the release of botulin. Now, I'm not sure how they do botulin toxin. Now, I'm not sure how they do this. But if you recall, botulin toxin is the same chemical that's used in Botox, and that stops you from looking wrinkled by essentially causing a neuromuscular blockade over the muscles that are causing a stretching of the skin. In terms of resistance, some bacteria can produce a conjugating enzyme, and that conjugation of the aminoglycoside means it's cleared faster through the kidneys. So just be aware of that. Next drug we're going to talk about is the tetracyclines. So like all the other bacterial translational inhibitors, they are bacteriostatic. They have a good broad spectrum of activity, but are particularly good against the atypical bacteria, such as chlamydia and mycoplasma species. And there's a case that comes up in the biomedical science exam about atypical pneumonia. We'll talk about it a bit more in the next video. But tetracyclines are one of the options for that disease, atypical pneumonia. Doxycycline is the main tetracycline that's used in clinical practice. You might have heard of it, you might have been prescribed it. It's also the prophylactic drug given for malaria if you're going to a country in South America, for example. In terms of pharmacokinetics, so this is what leads to the main side effects of the drugs. It's actually, the tetracyclines are chelating agents. And what's a chelating agent? It's essentially a chemical that binds ions such as calcium and magnesium. So the side effects, predictably, and the reason why these drugs are contraindicated in children and in pregnancy is that they can bind the calcium in teeth, causing tooth enamel dysplasia, bind the calcium in bone, causing reduced bone growth in children. They also lead to phototoxicity, so if you're prescribing this drug, you'll tell your patient to try not to stay in the sun for too long or to wear lots of sunscreen. They can cause gastrointestinal distress, like a lot of other drugs, and they can cause vestibular dysfunction. Bacteria have developed resistance to tetracyclines through the development of efflux pumps. So this is essentially a pump in the bacteria, and that just pushes the tetracycline out of the bacteria. And this pump, it doesn't just work on tetracyclines, it can work on a number of other antibacterial agents. So this is one of the mechanisms of multi-drug resistance bacteria. A drug that you won't see as much, so I guess don't have to know as well, is chloramphenicol. But it is a bacteriostatic agent with a good broad spectrum. It is good against sepsis and meningitis. And in terms of pharmacokinetics, as you can imagine, because it works well in meningitis, it's got a good tissue distribution and a good penetration of the central nervous system. To be cleared, it actually requires glucouronidation. So if you remember back to the bilirubin cycle, bilirubin goes from unconjugated to conjugated bilirubin through glucouronidation, through the enzyme glucouronyl transferase. Try to spell it. Uronyl transferase. And so if you have liver disease and aren't able to glucouronidate your drug and clear it, you need to reduce your dose. And the same in neonates. You might remember breast milk to neonates contains an enzyme which actually inhibits glucouronol transferase, and that's why you've got to reduce your dose. It's also an inhibitor of cytochrome P450. Now, if you remember, cytochrome P450 is a hepatic enzyme that is important in the metabolism of a lot of other drugs. So this can cause a lot of drug-drug interactions. So the main side effects is that it's got a dose-dependent bone marrow toxicity, right? And the other one is this thing called gray baby syndrome in neonates. And what's gray baby syndrome? Gray baby syndrome is this syndrome that comprises of vomiting, an ashen grey colour of the skin, limp body tone, hypertension, cyanosis, and cardiovascular collapse. And the reason that happens is because the drug isn't glucouronidated, as we said, because of the breast milk inhibiting glucouronol transferase, and you get this toxic accumulation of metabolites causing this gray baby syndrome. It's a classic question. It hasn't come up in biomed, but they seem to like it, especially in the USMLE. Okay, so the next group of drugs, and a very important set of drugs that we use all the time, are the macrolides. And these are good broad spectrum antibiotics, and they have good activity against gram-positive cocci, but also atypical organisms such as chlamydia, mycoplasma, ureaplasma. And again, like the tetracyclines, they come up as good options in the atypical pneumonia case. They also have good activity against Legionella pneumophilia, Campylobacter jejuni, and H. pylori. Examples of macrolides, which I'm sure you've heard of on the wards: erythromycin, azithromycin, particularly in kids, and clarithromycin. There's also roxithromycin. So yeah, you see these drugs being used all the time because of their spectrum of activity. In terms of pharmacokinetics, again, good to remember, if you can, that they're a cytochrome P450 inhibitor, except for azithromycin, so they have a lot of drug-drug interactions. Now, the side effects can be covered by the acronym the mnemonic MACRO. And they include motility issues because they actually stimulate endogenous motilin, and that initiates peristalsis, so it can cause a bad GI upset. Can cause arrhythmias, particularly a prolonged QT interval, cholestatic hepatitis, rash, and eosinophilia. Right? So that's a good mnemonic to remember the side effects of macrolides. Clindamycin. So clindamycin is quite frequently thought of as a macrolide, but it is not a macrolide. But you should remember it has the same mechanism of action and the same mechanisms of resistance against it as macrolides. But I think the key thing to remember about clindamycin is that it has particularly good activity against Staph aureus, and it's actually the drug of choice for anaerobic infections above the diaphragm. And for that reason, it is particularly good for aspiration pneumonia. So that's where it comes up clinically. The other place it does come up is that it actually has very good penetration into bone, right? So it has good clinical value in osteomyelitis. As you also know, osteomyelitis is quite most often caused by Staph aureus, so it gets concentrated in the bone and can work well against the Staph aureus. Again, the side effect, as you've seen with a lot of the cell wall, the bacterial wall inhibitors, is it acts against the endogenous gut bacteria, which can lead to a pseudomembranous colitis secondary to an opportunistic infection with Clostridium difficile. Now, linezolid, you don't need to know too much about. It doesn't come up too often. It is a down-the-line agent for the treatment against vancomycin-resistant Staphylococcus aureus, vancomycin-resistant enterococci, and drug-resistant pneumococci. Has a side effect profile of bone marrow suppression. That's probably all you would need to know about this drug. Right, so the next big group of drugs in terms of antibacterials, we've talked about the cell wall inhibitors, we've talked about the bacterial protein synthesis inhibitors. These are the antibiotics that inhibit the synthesis of nucleic acid. If you recall, nucleic acid is involved in the replication of DNA, producing new DNA strands, and that's necessary for bacterial replication. So if you inhibit the nucleic acid synthesis, you inhibit bacterial replication. Now, within that group of nucleic acid synthesis inhibitors, there's two main classes of drugs. There's the drug class that inhibits the synthesis of folic acid, and there's the drug class that inhibits the synthesis of the nucleic acid directly, right? And let's get into this because this is quite a meaty topic. I'm going to actually come back to this slide and show you this. This is actually a simplified pathway for the production of folic acid, okay? And as you can see, there are two main enzymes that are involved in the production of folic acid in bacteria. There's dihydroteroate synthase and dihydrofolate reductase. Just remember those two enzyme names. And the other thing you need to know is that dihydroteroate synthase is not present in humans. It's only present in bacteria. So if you inhibit this enzyme, it's not going to cause any effects on the human production of folate. However, dihydrofolate reductase is present in humans. So inhibition of this enzyme is going to cause inhibition of folic acid synthesis in us. Now, there's two main drugs that inhibit folic acid synthesis. The sulfonamides inhibit dihydropteroate synthase, and trimethoprim and pyrimethamine inhibit dihydrofolate reductase. Okay, so this is all this slide here says. Let's go over it slowly. Folic acid is required for nucleic acid synthesis, which is required for replication and bacterial replication. As I said, sulfonamides inhibit dihydropteroate synthase. This is an enzyme not present in humans, and hence this drug has no cross-reactivity with our folic acid synthesis. Trimethoprim and pyrimethamine, however, do inhibit dihydrofolate reductase, which is present in us, and there is cross-reactivity. Unfortunately, sulfonamides are not administered alone in the use because of multiple modes of bacterial resistance. Hence, the two drugs are actually given together, and they work synergistically to decrease resistance. Let me show you some clinical uses of folic acid synthesis to demonstrate this. So, you know what? I'm going to leave this talk about sulfalazine for another talk when we talk about inflammatory bowel disease. Let's stick to the antibacterial actions of these folic acid synthesis inhibitors. So, the drug that you'll see very often in clinical practice, so anytime you see a drug often in clinical practice, it can definitely come up in your biomedical science exam, is this combination of trimethoprim and sulfamethoxazole. So trimethoprim inhibits dihydrofolate reductase. Sulfamethoxazole is a sulfonamide, inhibits dihydropteroate synthase. And that combination of drug is known as Bactrim, right? So you might have heard that. It's prescribed a lot. And it's prescribed a lot because you have a broad spectrum of action, and it's bactericidal for gram-positive and a number of gram-negative bacteria such as E. coli, Shigella, Salmonella, H. influenza. You might know that it is a first-line treatment for UTIs. It's also a backup for amoxicillin in the treatment of nocardia and listeria, and also used for prophylaxis and treatment of fungal pneumonia secondary to pneumocystis jirovecii in HIV patients. Another combination that's used in clinical practice is pyrimethamine. That's your dihydrofolate reductase inhibitor, and your sulfonamide sulfadiazine, so dihydropteroate synthase inhibitor. And that's used for the prophylaxis and treatment of toxoplasmosis in pregnant women who might have been exposed or have contracted toxoplasmosis, and HIV patients with a particularly low CD4 cell count, below 100. I hope that makes sense. Pharmacokinetics-wise, it's important to know that these drugs are conjugated before they can be excreted renally. They have a high protein binding tendency. So in neonates, for example, if you bind all the albumin, which is necessary for their unconjugated bilirubin to bind to, you can cause a high unconjugated bilirubin count, which can lead to kernicterus. So just be aware of that. Now, the side effects. As I said, sulfonamides work on dihydropteroate synthase. That enzyme is not present in humans, so that doesn't cause any problems. However, because it is a sulfur-based drug, it can cause hypersensitivity reactions such as Stevens-Johnson syndrome. This is a very feared complication. It's also known as toxic epidermal necrolysis. Look it up on the internet. It has some horrific pictures. It is a side effect you do not want. It can lead to hemolysis in G6PD deficiency. Can also lead to phototoxicity. Right? Now, trimethoprim, pyrimethamine, they actually inhibit our own folic acid synthesis, so it's understandable why they suppress our own bone marrow. And this is why it is contraindicated in pregnancy because of the need, for rapidly replicating cells. Which takes us to the other drugs in this class, which are the direct inhibitors of nucleic acid synthesis. So that includes the quinolone group, right? The quinolone group works by interfering directly with bacterial DNA synthesis by inhibiting topoisomerase 2 and 4 includes drugs such as norfloxacin, apologies for the spelling mistake, and ciprofloxacin. Activity and clinical uses, so it's got a range of activities, these drugs. Again, sorry, I should have mentioned before that the quinolones, again, they don't really come up too much in the biomedical science exam, but good to know of them. Any of you who've travelled to overseas countries, it's often given to you by your GP before you go, just in case you get a bad traveller's diarrhea. But it does have a few other clinical uses, such as urinary tract infections, second line after the use of Bactrim, sexually transmitted diseases and pelvic inflammatory diseases because of action against chlamydia and gonorrhea, and they all work by a one-time single dose. They have particularly good action at skin and soft tissue and bone infections when gram-negative organisms rarely cause them, and they're good against drug-resistant pneumococci. But again, you should know of this, but they're not drugs that you use all the time, right? In terms of their pharmacokinetics, they actually bind really well to iron and calcium, and hence if they bind like that, it actually limits their absorption across the gut wall. So the instruction you should give if you ever prescribe a quinolone is never to take them with a meal. They're eliminated by the kidney, and like all drugs that are renally excreted, if there's renal dysfunction, you should reduce your dose. The side effects include GI distress, phototoxicity, rashes, tendinitis and tendon rupture, seizures, contraindicated in pregnancy, as you can imagine. You need rapidly replicating DNA, and cause a long QT interval. Now, that's a long list of side effects, so try to remember as many as you can. But again, like I said, I doubt you'll be asked about this particular class of drugs in your exam. But good to know about, has a lot of uses. Which takes us to our last and small unclassifiable drug, metronidazole. Mechanism of action is not fully understood. It is bactericidal. Its spectrum of activity includes antiprotozoal. It's antiprotozoal, so it can be used against Giardia, Trichomonas, Entomoeba. Also antibacterial activity, strong activity against most anaerobic gram-negative bacteria and Clostridium species. You should know that side effect of metronidazole is that it can taste metallic in the mouth, and it's got disulfiram-like effects. So if you remember, disulfiram is associated with a severe flushing and a terrible impending feeling of doom. Apparently you get that with metronidazole as well. And just so you remember, the main take-home message, we use metronidazole against anaerobic gram-negative bacteria. Okay, so that's all the antibiotics that I think we need to cover to prepare you for the biomedical exam. Now, I do know there was a lot of drugs that we covered in these two videos, and so I just want to say something about that. You don't need to know absolutely all of them. I think, like a lot of things that are being tested in this exam, you're going to be tested on principles, you're going to be tested on your rationale, and I hope that we can pull everything together and talk about what's really, really important clinically from this antibiotic topic in the third video, where we're going to look at the most clinically important infections that you're going to see. So your meningitis, so your urinary tract infections, your pneumonias, community-acquired atypical, your bacteremia secondary to, through bloodstream infections. Those things that you'll see quite often we're going to cover. We're going to see what the main causative organisms for those infections are, and we're going to see what the antibiotic regimens are for them. And then you'll know from those examples what antibiotics you really need to know well for your exams and for clinical practice. So thank you for watching the video. As usual, any comments, feedback, greatly appreciated. Any questions you might have, feel free to message me, catch up with me at hospital. Happy to catch up with you anytime. Thanks very much.